Cystic fibrosis is an autosomal recessive disease, or in other words, a mutation from obtaining recessive genes. In short, if both parents are carriers as in they have one copy of the CF gene, then they have a 25% chance of having a child with both copies of the CF gene. Now to explain the cause of cystic fibrosis, we have to go over the function of the protein that is affected by this mutation from obtaining both recessive genes. The part that is affected is the CFTR, which is Cystic Fibrosis Transmembrane Conductance Regulator. Its function is to diffuse chloride ions in and out of the cell. The signaling cascade for this function starts by a ligand called CGRP, otherwise calcitonin gene-related peptide, that is stored in the nervous system. The ligand bonds to a G protein coupled receptor, which then phosphorylates a GDP molecule on a G protein. The G protein then activates adenylate kinase that then converts ATP to CAMP, the second messenger that then activates protein kinase A. Protein kinase A phosphorylates the R group of the amino acid chain, causing interaction with the N-terminus, the starting point of the polypeptide chain. The interaction allows two halves of NBD and nucleotide binding domain to attract an ATP molecule and close. The presence of ATP causes the CFTR to open and chloride anions to enter and exit through the pore. Once hydrolyzed, the ATP molecule leaves the CFTR and the channel closes. How the CFTR affects this cascade is that the mutation causes the CFTR to not work or exist at all. In lower cases, CFTR instructions will be sent from a DNA as mRNA to the rough endoplasmic reticulum, where the ribosomes will synthesize and transport to the plasma membrane. The mutations in the gene chromosomal 7, which contains instructions for building CFTR. The change in the gene is called delta 508. Delta 508 means deletion of phenylalanine on position 508 as 3 letters on the amino acid code are missing. This makes the CFTR shorter, which the cell quality control system sees as an error and flags it for destruction. With CFTR missing, sweat is more saltier since in the gland where sweat is, there is no chance to reabsorb the chloride ions in it, which causes the body to lack chloride. Also, in the pancreas, with the CFTR missing, the pancreas cannot make the slurry mixture to protect themselves from digestive enzymes. Normally, chloride is transported in and out. When more chloride is added, the water potential is lower with more solute. With lower water potential on the outside of the cell, the water will diffuse out of it. Water diffusing out of the cell will allow pancreas to have the slurry mixture. Yet with the CFTR missing, the chloride ions can't get out, leaving no chloride to be added to have a lower water potential on the outside, making water not diffuse, which means that the pancreas will not be able to have the slurry mixture. With no slurry mixture to protect them, the digestive enzyme will not digest the pancreas, causing inflammation. In the lung with no CFTR, there is thick mucus in the areas that cannot be swept out, as the CFTR will not be able to let chloride out. This makes the inside of the lung cell have a lower water potential because of more solute and have the water cross the membrane into the lung cell, leaving the mucus to be thick and less diluted. Mucus then builds up in the bronchioles and reduced surface areas are available for oxygen exchange and the bacteria, causing more infections. The body's immune cells will release enzymes to kill the bacteria, but incidentally kill lung cells, causing chronic infections. Where the stomach and small intestines connect, digestive enzymes pass through to digest food, but with a lack of CFTR will also create mucus there and clog enzymes from entering and causing malnutrition as fat cannot be absorbed now.